In the previous video, we studied about what is gene cloning, and now we will see how we can actually transform the cells and how we can select the propagation of cell clones. So we saw that we have firstly this is the cloning vector. So this is the cloning vector, and for example here this is the target DNA. So here we have the target DNA. So after the insertion we get here the recombinant DNA molecule. So here we have the recombinant DNA molecule. Now this recombinant DNA molecule so we call it as the chimera. So this is the chimera, and we will be having also the non-recombinant vector. So this is the non-recombinant vector. So here we have the recombinant vector and this is the non-recombinant vector. Now then we introduce cloning vector into host cell. So when we introduce the cloning vector into host cell, so here in the host cell we have here this is the bacterial chromosome, and we have also here that is the recombinant DNA that has been inserted into the host cell, and this is the transformed cell. So this is actually the transformed cell. Now we have also the non-transformed cell. So in case of the non-transformed cell, what will happen? So in the host cell we will be having only the chromosomal DNA. For example, in case of this bacteria we have only the bacterial chromosome, and this is the non-transformed cell. So this is non-transformed cell. Now among the transformed cells in which our vector has been inserted, we have two types. One is the transformed cell having the insertion of the recombinant vector, and one without the recombinant vector. So the selective propagation of cell clones, so here this selective propagation of cell clones, it involves two stages. So initially the transformed cells, they are plated out by separating on an agar surface. So they are initially the transformed cells, they are plated out. So they are plated out by separating on an agar surface, so that we encourage the growth of well separated colonies. And these are cell clones or populations of identical cells that are descended from a single cell. And then subsequently, individual colonies can be picked from the plate. So, individual colony can be picked from the plate, and the cells can be further expanded in the liquid culture. So, these can be expanded in liquid culture. Then, the isolation of recombinant DNA colonies. So, this isolation of recombinant DNA colonies by harvesting expanded cell cultures and selectively isolating the recombinant DNA. So, in this way, we have here the recombinant DNA clones. So, the clones are basically the recombinant DNA clones. So, we will get here the recombinant DNA clones. So, these are all the recombinant DNA clones.